I'll be your host today. I'm Helen Taylor-Boyd. I've been working with Bats Without Borders for a while now, and I'm also on the board for Bats Without Borders. For those of you who don't know very much about us or have recently joined our Winged Wednesday webinars, we are a Southern African NGO working on projects across Southern Africa on bats. We do research, applied research, engagement, capacity strengthening, and evidence-based conservation strategies. You can actually find out more about us on our social media channels as well as our website. Be aware that we are no longer on Twitter, so we're on Blue Sky, so you can find us there. You can also join our WhatsApp group, which gives you basically the latest updates. There's a QR code on the bottom right hand of the screen right now. I'll leave it up there for a couple of seconds. Hopefully we'll have a flurry of new WhatsApp group members. So please do sign up to our WhatsApp group there. Also, these Winged Wednesday webinars do cost to run, and we would like to keep these going for the longer term. So if you are able to support us with a donation, there is a QR code to the link to the donation page there. I'll also pop it in the chat in a bit. So yes, any support you can give would be wonderful. Also on our website and social media channels, you'll see some Halloween campaigns that we'll be running over the next week or so. So if you're able to support those, that would also be great. Couple of updates from us before we get on with the webinar. We have a Classroom Without Walls program running, and we are delivering Train the Trainer sessions. If you are interested, we do need you to be a trainer that is based in Southern Africa. If you would like to sign up for more information, there's a QR code at the top there. So do follow that to enter your details to sign up for more information. And then we're also excitingly running our first formal community science project using an app to collect data on fruit bats, so for a fruit bat monitoring program, and that's being launched as we speak. You can sign up for more information on that using the QR code there as well. Hopefully you've had enough time to sign up to that through the QR code. If you haven't had enough time, don't worry. Obviously, you can watch back on this Winged Wednesday webinar to find the QR code, or you can find more information on our website as well. There is also a webinar running at the end of November when we'll be hearing from Pipat, who will be talking about an endemic leaf-nosed bat. So please pop that date in the diary. A quick reminder for those in the UK, for example, where times will be changing, so your clocks will be changing. Please watch out for changing times of the session in the area that you're in. And today, we'll get on with the Winged Wednesday webinar from Laura Torrent, who's with us today. She's worked on bat research and conservation across Europe and Africa since 2013. She's combined bioacoustics and ecological surveys to study bat diversity, human-wildlife interactions, and forest conservation. Her earlier work focused on hibernating bats in Poland, and then followed by international projects in Mexico, Madagascar, and the Amazon. But for the past 8 years or so, she's been focused on the bat fauna of Equatorial Guinea, which has led her onto her PhD with the University of Porto in Portugal, also with other collaborators in Portugal and Spain. And for this, she's been exploring the taxonomy and ecological roles of bats in Central Africa. She also collaborates on global bat conservation efforts and contributes to IUCN assessments. But today, Laura will be presenting an overview of her research on Equatorial Guinea's bats. And that includes the first comprehensive update of the country's bat fauna in decades, and important clarification of taxonomic uncertainties. There's some particularly exciting news that she's going to be sharing with us today. I won't spoil it. So without further ado, I will hand over to Laura.
Thank you very much, Helen. Thank you everyone for being here, and especially to Bats Without Borders for inviting me to these lovely events. I'm a big fan, so it's an honor to be hosting one. I need to apologize to everyone because I'm sick, so unfortunately I'll be coughing at some points and my voice is not ideal, but I'll do my best to keep up with the presentation. So let's move on. I'm going to start talking about my early experiences, how did I end up in Equatorial Guinea. So this is a young version of myself from 2018. Very excited, taking pictures of Mionicteris torquata with my camera for the first time. And everything began because I had a friend, Diogo Ferreira, that some of you might know. And he just told me, hey, Laura, I'm going to Equatorial Guinea for an expedition and I'm going to be looking for bats in the jungle. Do you want to join us? And I was like, hell yes, of course. And it just happened like this. And well, Diogo moved to his project and I just got hooked with the country and I keep going for the years. And this is how my story started, just with a kind of easy introduction from a friend of mine to an easy invitation, let's say. But where is Equatorial Guinea? So from now on, I'm going to call it EG because it's shorter and it's easier to pronounce. EG is found in Central Africa. It is found between Cameroon in the north and Gabon in the south, and is divided into regions. It has the insular region, which is combining several islands. Bioko Island is the largest one, and then we have the mainland region. So most of the landscape from Equatorial Guinea is lowland rainforest. Sadly, in the past 3 decades, the situation has been changing quite dramatically. So habitat fragmentation due to urban development, road constructions has been logging and altering the original rainforest. And of course, this is causing a major loss of biodiversity. The problem is that we don't really know what we are losing because there is not much research going on in EG. So one way to try to tackle this is to actually do surveys on very diverse groups, such as rodents or bats. But how many species can we find of bats in EG? That's the question that I asked myself when I was looking into literature in 2018, excited to go to the field. And I was surprised because there were not that many publications. So the most recent publication of bats from EG is from 1994. So it's just 2 years younger than me. And it was a paper that focused on the bats from the islands, not even the mainland part of EG. If we want to know when was the last research done on the inland region of the country, we need to go back in time to the 70s, when Clyde John described 22 species for that region. And that's all. There is nothing else. There was just like this big void of knowledge. So of course, this gives us plenty of room to explore and a lot of questions that we could answer, but starting from the basic. So here is where I come up with these 2 big data sets that has been the 2 pillars of the work that I'm going to be presenting to you today. So on one hand, we have the museum collections, and on the other hand, we have expeditions that we have been doing from 2018 until this year. Let's start with museum collections. So the largest collection of bats from EG is found in Spain, in southern Spain, in Sevilla. It's hosted at the Doñana Biological Station, and it has more than 1,000 specimens. They are all prepared in different ways. Here you have an example in a plastic box where we can see all these specimens nicely prepared, labeled, and separated with their skulls and mandibles. So I've been sorting out all these specimens, checking their labels and checking their identities and updating it in case that it was wrong with the current taxonomy. Another part of the work that I've been doing with the collection from the Doñana Biological Station is actually checking the localities from each specimen, because most of the specimens had only written down the name of the village or the kilometer in the road between two main cities where it was captured, but there were no GPS coordinates. So that took me going to old maps from the area and looking for all these places and then finding the coordinates and yes, filling in all these basic and essential information. I've also been taking a lot of measurements from skulls that were already prepared in the moment that the animals were taken, but we have also taken new skulls from individuals that were not removed. And all these measurements, of course, give us key information that allow us to distinguish between cryptic species. And is essential information that we need when we are describing a species, either it's known or if it's new for science. Another element that I didn't know about it and I have discovered exploring this collection is the baculum. Some of you might have heard me talking about this before. This is a tiny bone that is found in the penises of many bats. And it has been proven to be very useful to distinguish among cryptic taxa. So in this case, for Pipistrelloid bats, as you can see here, that they have this very similar external morphology, this tiny bone is proven to be very, very useful to distinguish these species. So we've been extracting this baculum for many specimens and taking photos, measurements, and describing it. Another element that I've been developing while exploring the collection is the photography, because this collection is in a specific locality in southern Spain, and unless you get a permit and you have fundings to actually fly all the way to there, you have no idea that exists, you don't know what is in there. So I've spent a lot of time taking photos of all these specimens, hoping that this can be useful for people in the future that needs to have a look at some species and specifically, or just have it more accessible to everybody. So of course this has been a learning process. And at the beginning I was just using my dish scoping technique, so the phone, and then you had the magnifying from the ocular from the magnifying, and I was trying to take pictures this way. Of course it worked. I could actually see the skull, I could actually see very well the teeth and take some photos, but maybe it's not the ideal kind of pictures that you would like to post in a scientific publication or for a presentation. So I've been improving my skills and asking advice from a lot of people, and nowadays I'm able to take these pictures. And here I just wanted to briefly show you how I do it. So I take a picture of the number, the label of the specimen, so this way I know which individual it is and the species. And then I just move the position of the skull depending on the angle that I want, using this tiny plaster that I try to hide as much as possible. And this is a way to do it, but it can be done in many other ways. But I find it quite easy now that I've been practicing a lot. So moving on, let's go to the second pillar of our dataset, which are the recent expeditions. We have surveyed bats in more than 60 localities from Bioko Island and the mainland region of Egypt. We have surveyed all kinds of habitats, from urban areas to primary forest and all kinds of roosts, from like a log or even artificial roost, like could be an abandoned building. Mostly we have been using mist nets, usually ground mist nets, but whenever we could, we try to put them as high as possible. I'm pretty proud of this one, because here we were able to stack together 218 mist nets, and they are more or less 5 meters above the ground. And although we didn't catch anything until the last minute of the night, that we were already super bored and wanting to go bed, and my colleague is like, hey, there is a leaf on the top of the net. And I was like, it can't be a leaf. I mean, there is no wind. And then we just realized it was a bat. So we were just going as hell, going super fast and trying to get that individual. And luckily we got it. And this happens a lot. So most of the time we would just get very few individuals in the nets, even in the hard traps that we use in the last 3 years only. But every individual was a different species. So low numbers, but different species. And sometimes we didn't even had a clue of what was that species that we had in our hands. So this led me to the first chapter of these presentations, which are the Pipistrelloid bats. So this group of cute brown bats, it is a nightmare. They are really difficult to tell apart. So despite there has been a lot of progress in the past decade, thanks to a lot of work combining morphology, genetics, and new specimens that has been able to untangle the situation with many Pipistrelloid bats, there is still some situations that are not well resolved, and there remain taxa that is not clear to which genus they are part, or it's really still a work in progress. So for this project, we wanted to update the systematic of African Pipistrelloids, and in this case, we wanted to focus on the Pipistrelloid bats from the Congolian rainforest using data from EG, and also resolve the taxonomy of 2 cryptic taxa that we had in our hands already. So for that, we've mostly been using morphology and genetics. Here, to resolve the morphology of 2 cryptic Pipistrelloids, we have used a principal component analysis, where we can see in the morphospace how the different skulls are distributed. So this is the size of the skull, and this is the flatness. And we can see that here we have Musiculus, which is the smallest one because it is on this side of the x-axis, and this is Hapoldoron, which is the largest one of the species that we have studied. And then on the y-axis, we have the flatness. So we have that Crassulus is the species that had the flattest skull compared to all the species that we studied. So this gives us some information already on the skull morphology of the Pipistrelloid bats from EG. Then we also had a look at the Crassulus and Musiculus baculo. And as I told you, as you can see, these are different species, and you can see this is clearly being shown by the shape of the baculum. They are all completely different size, different shapes. It's very remarkable. Moving on to genetics, we used cytochrome and we did a phylogeny with more than 40 different species. And I'm going to make it easier for you. I'm going to walk you through this long phylogeny. So this is where we have Crassulus. So we confirm that Crassulus is indeed a different species from Bilieri, and both form part of the genus Afropipistrellus. There is like a 16% of genetic divergence between Crassulus and Bilieri. So we confirm it. And secondly, for the first time, we know that Musiculus indeed is not part of the Ipsugo genus, but actually as the Pipistrellus as well. So with this approach, we've been able to describe 11 Pipistrelloids for EG and expand the known distribution of 6 species all across Africa. But now I would like to focus on Bioko Island. So here we have a zoom where we have the peak areas, which are the white areas. So we were surveying bats at almost 3,000 meters above sea level, and we caught this tiny pipistrelle that we weren't sure where it was. But it resembled a specimen that my supervisor and his colleagues collected in the 90s in another mountain from Bioko Island in Lago Biao. So who is this guy? We decided again to do a morphological analysis using a principal component analysis. And here we can see that, again, the largest skull is on the left side. It's the opposite on the graph that we saw before. And this is the Pipistrellus from Bioko Island. So this guy is the largest one. Then we check again the morphology of the baculum, and we see that the Pipistrellus from Bioko Island, although has a similar baculum to other Pipistrellus from mountain areas, it is different. Finally, we also did a phylogeny with cytochrome B molecular marker, and we can see again that they make a very nice clade separate from the rest of Pipistrellus. So these morphological and molecular evidences prove us that indeed we have a new species of Pipistrellus for the mountains of Bioko Island. And we name it Pipistrellus etula. Etula in the local language from the people that live in the island, that they are called Bubi. it means island or god of the island. So we thought it might be a nice name to give honor to the locals and the inhabitants of Bioko Island. And as a nice surprise, apparently Pipistrellus etula is the 1,500 bat species of the world. So that was an extra amazing result. And in case you're interested, you can have a look at the paper online here. So let's move on to the second chapter of this presentation. Now we're going to talk about Molossids. This photo is not from Equatorial Guinea, but I think it's really cool, and I thought it was nice for the for the start of this section. Molossids. Molossids is another interesting group. They have a quite remarkable conservative morphology, which makes sometimes it very difficult to tell them apart. Plus, there has not been like a full revision of the whole family yet, including all molecular markers like I don't know, mitochondrial, nuclear. This is not done. So this means that we don't really know exactly some genes exactly if they are well named or not. And these, of course, give us a lot of problems. For in Africa, it's not an exception. We know nowadays that there are more than 60 species, but yet some have only been caught once, and there is only the type specimen, and others just have very few specimens that have never been sequenced, so we don't have much molecular data to actually use them for phylogenetic analysis. So it's another interesting and complicated group to explore in Africa. So this was in 2023. We were surveying bats in this protected area from mainland EG. It's called Piedra en Sas, which is a national park, and it has a primary forest that has not been logged. And here, so we set usually the ground nets, and we attempt to set a canopy net, which although we had some difficulties, we managed to install it. And at some point of the night, my supervisor just comes to me like, Javier was like, oh my God, we have it. This is a new species for science, for sure. It was this tiny guy. So he was like, oh, I've never seen something like this. This must be a new species for science. I've only seen them once on one of the islands from the Gulf of Guinea. So we were all very excited. Of course, we got the permits and we exported this specimen to compare it with the specimens that Javier was referring, which is Mopstomensis. So this species was described by him and colleagues from the island of São Tomé, which is found in the Gulf of Guinea. And it was only described based on 3 females. So here we have our male from mainland EG and the 2 females from São Tomé Island. So we were like, are they the same species? I mean, look at them. The size of this male is quite small compared to the females, and the females had a completely different coloration. And then again, the situation with the distance. I mean, Tomensis is supposed to be an endemic species from São Tomé Island, whereas this species was found on the eastern side of mainland EG, close to Gabon. So could it also be the species that is only known from one specimen, Galageri, found from Congo in the 70s? We didn't know. I mean, there were too many questions, so we thought it might be nice to actually do a morphological and genetical analysis of this group of molluscid bats. So for that, we first need to find more information on Galageri. And this is all the information that we actually had from Mops Galageri. These beautiful drawings from the 70s, of course, they are really good, but to compare our pictures with this was a bit hard, and there was only one specimen on top of that. So I asked permission to the Museum of Natural History in London, and I went to visit the collection and actually take photos and measurements of the holotype of Galageri myself. So these are the first pictures of Mops Galageri. It has this unique pouch that has these 2 of hairs that come out. It's a bit like Chapini that you probably know. And it has this is the lateral side of this pouch that opens. So then we compare our individual from mainland EG with Mops gallagheri. And although they had similarities in the shape and that they all share this tube of hairs, we could probably starting to see that they are indeed different stuff, different taxa. Here you can see some details from the frontal part of this pouch or interaural lobe. This is another word that we can use. And we can see that the Molossid from mainland EG is quite similar to the females from Sao Tome, and it's quite different from the male gallagheri, the only individual known from this species. We also compare the tragus. So here we can see that Mops from mainland EG and Tomensis could be quite similar, but then we see different shape for gallagheri. And here I couldn't help but show you a tiny video of how we actually do these kind of photos. So we need to be creative. And this is a colleague of mine from Doñana Biological Station, and he kind of fixes the individual without pinning anything on the specimen. We just block it, and then we put a piece of paper behind the tragus. So this way we can take photos that then can be easily comparable with the other specimens. Moving on, we also, of course, compare the skulls. And here we can starting to see some remarkable differences. This is, again, Mops from mainland EG, Tomensis, and gallagheri. And although they had differences, it was not that clear that actually the Mops from mainland EG was different from Tomensis. What we could actually see is this frontal part of the skull. It is kind of inflated, and this is a unique feature that we can find only in these three species, and is not found in any other African molluscid, but along other morphological details like in the teeth and the skull and so on. So this kind of started to give us hints that this might be a new subgenus for the group of molluscids. So we, of course, needed some molecular support. So we attempted to extract DNA from Mops tomensis, which were the specimens from Sao Tome Island. And this wasn't easy because these specimens were preserved in formalin. And this is a technique that degrades the DNA. So then it makes it really hard for us to actually amplify long fragments of DNA. But after several attempts, we managed to get it. And surprisingly, we found that Tomensis from Sao Tome is actually the same species as the individual that we have from mainland EG. It had only 1% of differences between the two individuals, so the ones from the islands and the ones from the mainland, which is quite surprising considering that the island of Sao Tome is more than 500 kilometers away from the mainland in the area where we caught the Tomensis from the mainland. So this just raises more questions about these species. How much do they fly? Are they able to actually cross the sea, although they are small molluscids? Probably yes. Is this still happening? How is it that it's so difficult to actually catch these bats? We don't know anything about the ecology of these species, although we know that nowadays we know it's not endemic from Sao Tome Island, but actually it can be found in mainland Africa. So based on the morphological features that are so unique for this group, we decided to describe a new subgenus for this group of molluscids that we name it Ornatomops, which means ornament, making reference to this big piece of skin that opens and has this hair that opens like a fan. And here, in case you're interested, you can also have a look at the publication. And this is only a group of the molluscids. As I mentioned earlier, there are many other molluscids, especially the ones found in the forest that are still very rare, very few individuals that are known. So there's plenty of more work that can be done attempting to resolve the taxonomy and systematics of Molossids, not only from Africa, but maybe even for other parts of the globe that are not clear yet. But let's start attempting for Africa so far, which is still pretty challenging. And so on. Now we're going to move to the last section of my presentation. So here we compile basically all the localities from which we have specimens, or that we've been surveying bats, or localities that were mentioned in these very old publications previous to our work. And as we can see, it covers quite well the entire mainland region of EG. So the checklist that we have prepared is only focused on the mainland part of EG. And we can see here the protected areas as well, marked in light green. So we have surveyed more than 100, well, we have more than 120 localities that you can see here, from urban areas, again, inside protected areas, quite well distributed along the landscape. And if we compile the specimens that I have examined and the bats that we have collected or that we have surveyed along these years in recent expeditions, we have here 1,100 specimens examined and almost 700 individuals that we have caught, which, yes, it's not that many individuals, but as I told you earlier, it's very difficult to actually catch these bats. So before our work, we knew 25 species, and after these recent expeditions and this thorough review of the specimens from the museum collection of Doñana Biological Station, we have more than double the number of bats found, the number of species known for mainland EG. We have a species like Glauconicteris superba, which is a very rare species, only known from 5 localities all across Africa. So now we know another country in which we can find this species. So this data can give us valuable baseline information for species that are really hard to actually survey. And also, again, genetics, just by getting wing punches and using cytochrome, we can actually confirm the identity of some cryptic species like Scotonicteris bermansi, which is a tiny fruit bat. Just to give a bit of more details, here we can see the different families of bats that we found in mainland EG and how they are distributed, the number of species, based only on the data from the expeditions from 2018 to 2025. And here, if we include the data from the museum collections, we see that almost 30% of the species have only been found thanks to exploring the data that has been collected and stored in collections. Especially for Embalaneuridae family, we could only actually have proof of the existence of these species on the areas thanks to the specimens that were collected in the 80s. So we have 33 new species records for mainland EG, but although most of them have been confirmed, we still have like 3 that remain unresolved, and they require further morphological and molecular analysis and probably more specimens and more data to actually untangle the situation with these taxa. So again, plenty of work that can still be done with Central African bats if you're interested in taxonomy, or if you have collections that you can go back and re-examine those specimens. Finally, I would like to mention all the capacity building work that we've been doing along these years, mostly with our main partner in the country, which is INDEFOR, which is the governmental institution in charge of the protected areas and the protection of the biodiversity. And this is my colleague Esther, that she has started to learn 5 years ago with me, and she has developed her bat researcher skills, and I'm really proud for her because last year she was able to win her first grant to do her first study on bats in one protected area from mainland EG. And this is one example of one of the successful experiences we had along these years with the capacity building in EG. And also we were able to join forces with the University of Equatorial Guinea on Bioko Island, and we took some students from environmental sciences to join us and do an introductory course on the study of bats. So here we have one of our first interns, which was Pablo, and Pablo was already a bit advanced. So here in this case, he was teaching the students how to safely remove a bat. And for me, this is also a nice success and a necessary element and actions that we need to do all the time that we go to the field as well. So I've been talking a lot, so let me recap for you what could be like the big highlights of all that I've presented you today. We have described a new species for science, this mountain Pipistrellus etulo on Bioko Island. We have found Mops tomensis. This was supposed to be an endemic from Sao Tome Island, but now we know that it can be found also on the mainland. So we have expanded quite significantly the distribution of this rare Molossidae bat. And we have also described a new subgenus for this group of rare forest-dwelling Molossidae bats. And finally, we have more than double the number of species known for mainland EG, along improving the distribution of many rare taxa. And finally, I would like you to really take this as a final message, because I think it can be applied to other Central African countries. So collections really matter. If you have access to your museum collections, have a look at them again, take measurements, take pictures, and just review them again, because they might give us some hints to actually keep resolving some entangled situations with some groups of bats in Africa. Why? Because by naming a species, in this case describing a new species, we have the baseline information that then can allow us build up conservation actions that can be able to not only protect that bat, but maybe other species in the area. So that's why taxonomy and naming species matters. And finally, invest on local capacity and strengthening collaborations as much as possible, because we researchers can do a certain amount of work, but then the authorities need also to be engaged and be part of the project, and they are the ones that can actually ensure that this lasts for many years. So I feel it's also essential that we strengthen these three pillars to keep improving bat research and conservation in Central Africa. I cannot but finish thanking everyone. I'm very privileged to be here today presenting to you, but this has not been done by myself. It's been a massive effort with a lot of people that has helped me along the years in the field, in the labs, funding institutions that have supported my research. So I'm thanking all of you for your support, and yeah, and thanks to all of you for attending my talk.
Thank you so much, Laura. That was absolutely fantastic. And I love all your beautiful photos that you've included in your presentation. And you've got a few comments in the chat about your lovely photos as well. Actually, you know, having had a similar experience with my PhD working in an area where very little is known about the bats in the area I was working in, it definitely resonates when we're thinking about the bat work across Africa being very limited and so much more still to do. So really exciting to see all the work you've done. And of course, your fantastic news. So we're up to what number of species across the globe?
1,500.
Woo!
Yes.
So very, very exciting work that you're doing there. So just to the audience, if you can pop your questions in the chat before you forget about them. If you would prefer to ask them out loud, that's absolutely fine. If you can pop your hand up and we'll get to you as soon as possible. So we'll just head back to the very beginning. There's quite a few fans here from all over the globe. I'm always impressed about you know, where everybody comes from on these webinars, joining us from all over the world. And it's, yeah, no different today, all over, you know, countries outside of Africa, and very exciting. So, our first question we have was about the radiography of the bacula. Can that be done in the field in any way?
You mean X-rays?
Yeah. So this was Tybalt asking this sort of question, about what do you do in the field, basically.
Okay, that's a good question. I also ask myself if that would be possible, but I've been asking around, and apparently it would be really tricky to actually do that. I mean, we are talking about like 2 millimeters, 5 millimeters. So first of all, where do I get this X-ray machine? In EG, it would be super complicated to bring a bat from the forest actually to a hospital and ask permission to actually attempt to do this. And I don't have the funding to actually have an X-ray machine with me. So ideally, that would be fantastic, but I can't do that. So what we've done has been basically with the specimens that we had already in the collection. So we basically cut the penises and we we extracted the baculum from those specimens. But what we are also seeing is that the baculum somehow is linked to the shape of the penis. So usually you could see some morphological differences from the penises that could give you a hint that we might be dealing with two different taxa. So yes, of course, we don't really want to collect specimens, but in some cases when we are dealing with so cryptic and unknown taxa, this is still necessary. And this is something that I've learned along these years with my own experience and talking with a lot of African researchers. So sometimes it is necessary because then we can find other morphological or genetic evidences that help us to actually describe that species, and then we don't need to collect anything else. And then we know that species, and then we can develop conservation strategies. So
yeah, definitely highlights the importance of specimens, doesn't it? You know, especially when we're working in areas where little is known. I suppose my question later on, I had asked links to your comments about how you know, the bacula can link to physical characteristics that we can see in the field. So my question was actually about the inflated muzzle area. Do you have any thoughts on what that function might be in these particular molluskids? Or is it just a characteristic that we literally don't know anything about?
I feel right now the answer is more like we don't really know right now. I mean, we have 5 specimens known from this new subgenus from Ornithomobs only in all Africa. So it's really hard to actually, I don't know. It's really hard to actually thought of what it could be used for, any reason or not. Maybe it's just a morphological thread related, a feature related to a different sex, because of course we see that in males it is a bit more conspicuous, but again, we don't really know. It's still an opening question. We need to know more about Tomensis and Gallagheri.
Yep. So we need more manpower basically to do some more work to look at all these different aspects. We have lots of lovely comments for you, you know, saying that the talk was great and the presentation was great, photos were great, and also just complimenting you, Laura, as well on all the work that you've been doing. I know exactly how hard this work is, and you've been doing this for quite a while now as well. So yeah, fantastic that you've stuck on it, and getting to this stage where you're getting new species, and also hopefully soon your PhD as well.
Yes.
I think that's all of the questions. Or just a response there from Tybalt. Would be wonderful if within the near future there's a small portable radiography machine developed for the field.
Yes.
There's lots of things we'd love to use in the field, and yeah, photos only go so far. So again, lovely comments there. We've also got another question, actually more of a personal question. What is the next step for you after your PhD? This is from Tybalt.
Well, as I've already been highlighting along my presentation, you see that there is plenty of work that can still be done. Now we've just kind of scratched the surface, and we have this baseline knowledge for mainland EG, but many more research can be done. We can actually aim for more ecological questions and keep reinforcing conservation actions in the country. So I would love to pursue a postdoc and continue working with Central African bats. That would be the next step.
Fantastic. Another personal question Alex is asking. Amazing talk, Laura. Easy question, but what is your favorite battle family?
Oh, okay. I love Pipistrelloid because they have given me nuts, but I think my favorite one is the panda bat, Glauconycteris superba. Yeah. When we saw it for the first time, it was just like, damn, this guy is amazing.
Yeah,
I
very
would say so.
exciting. Yeah, fantastic. My personal favorites are Molossidae, so I love that you've included them and obviously done some amazing research on them here. Fantastic. I think that comes to the end of the questions and comments here. Oh, one more. Oh my goodness, I absolutely do not know how to say this scientific name. Did you ever catch Mops aloysius Dia? I don't actually know this Mops, so go for it.
Yeah. Oh, thanks for your question, Mamba. No, this is a very rare Molossid bat, and we haven't got it. No, no. I mean, but again, it's so hard to catch Molossids. I mean, all of you that have been doing field work in Africa or elsewhere, you know how hard these guys are to catch, unless they like the roof of someone. So no, it might be there. I mean, my supervisor was surveying bats in EG for almost 10 years, and it never caught Tomensis on the mainland. So who knows how many more molluscids we are actually missing. So yeah, that would be really amazing to explore. We can go again.
Absolutely exciting stuff. Another question from Purity this time. What has your favorite experience or memory working in EG been?
Oh, Oh wow, okay, that's difficult. I mean, there are so many. I guess, yeah, Glauconycteris superba. The first time that we caught it, because we were just surveying bats in a small creek inside the forest, and we set some canopy nets like at 6 meters over the stream, and we were catching some nycteris and some other pipistrelloids. And then all of a sudden, I hear one of my colleagues that he's usually very calm and doesn't make many noises. Is very quiet person, and he's like, "Oh, hostia puta!" He's just swearing in Spanish, but he doesn't usually do that. And I just went there, and my supervisor Javier was actually removing something that was black and yellow, and he was like, "This," and this other guy was like, "No, what's that?" Everybody was so amazed, and we got 3 individuals in that net. So we were just quickly setting it up again, catching another one. So it was just like, crazy. I mean, you never know when this can happen because, as I was saying, we have very few catch every night, and then from time to time, you just you find this gem in the middle of the forest. So it's just like never know. So yeah, I would say this is probably one of my favorite experiences.
So very exciting. And following on from that, Anira is asking, where do we sign up for the next expedition? Smiley face.
Okay, let me first finish the PhD, and then I'll start looking for funding, and we can talk. I mean,
That's
this
it.
is also important.
It's great having these field experiences and everything, and it all comes with a lot of data analysis. And then also, obviously, for future work, the finances is always one of those sticking points, isn't it? Especially for research in Africa on bats. Obviously, it would be great if we had access to a lot more funding. We do actually have some more questions coming in, right? From Amantio, do you think the information you have obtained can be used to create a list of bats in EG?
Oh, definitely. No. The third chapter that I presented, the checklist, is gonna be published soon. And then we are aiming to actually do a book in Spanish of the bats from mainland EG that can be distributed among the institutions from the country so that they can actually use it in the field.
Fantastic. An example of applied research and evidence-based conservation as well, I suppose. So fantastic. I think we have come to the end of the questions. I don't see anybody's hand up. So if that's the last few questions, then I think we will close the webinar. Just a quick reminder, you can find out more about Bats Without Borders using the website and our social media channels. There's also the link in the chat to the WhatsApp group. You can also watch Laura's talk after this again, or if you've missed any parts of it, or you can pass on the link to those of those who couldn't have made it and would have liked to come to the talk. We will be sending out the link on our news channels or to the YouTube channel where the recording will be, so you'll be able to watch that later. Great. Thank you so much again, everybody, for attending. And thank you so much, Laura, for that very exciting presentation and sharing that exciting news with us as well. Thank you very much. Enjoy the rest of your day, everybody.
Thank you. Bye.
Bye.